So hi everyone, and happy New Year, and welcome to the first seminar of this new year. And as we were talking before, I think most of us see the light at the end of the tunnel. Uh, so I take great pleasure in welcoming Dr. Olusola Ajilore, or is he also popularly called as Olu, uh, as our speaker today. So Olu is an associate professor and associate head for faculty development in the Department of Psychiatry at the University of Illinois, Chicago. He graduated from Harvard with a degree in biology and then went to do his MD PhD at Stanford, where he studied the effects of stress hormones on the brain. He then joined the residency track here at UCLA, where he transitioned into neuroimaging of major depression. He also did a brief stint as an assistant professor here at UCLA in the Department of Psychiatry before moving to UIC and starting his own lab. So Olu wears many hats in his department. He's the director of the Mood and Anxiety Disorders Program, the director of the Clinical Research Core for the UIC CTS, or CCTS, the Center for Clinical and Translational Science. He's the Associate Residency Training Director and Associate Director of the UIC MSTP program. I guess both of them, which probably take most of your time in your schedule. So at UIC, he runs the Computational Neuroimaging Lab, Computational Neuroimaging and Connected Technologies Lab, along with Dr. Alex Liao, whom we also know very well here. So his research uses novel computational neuroimaging techniques and digital biomarkers to better track and treat neuropsychiatric disorders. And so just a brief digression here. He does many different types of interesting research. And one of the things he does is analyze mobile typing patterns. He calls them kinematics, and looks at how that affects the mood and cognition. I think they had a paper in Jamia last July about this. Hopefully he can talk to us about that in his talk today. So his talk today is going to focus on novel network analysis methods for looking at alterations in structural and functional connectome in patients with internalizing psychopathologies. So let's welcome Olu.
Thank you very much for the kind introduction and the opportunity to talk at UCLA. The roots of our lab between myself and Alex are at UCLA, and so I wanted to thank Sarah Chang for suggesting me as a speaker, and I also wanted to extend a special thanks to Catherine Narr and the folks in her lab who really helped me get started on this path of research when I was a very neuroimaging ignorant residency track resident way back when at UCLA. So I appreciate the opportunity to talk to your group. Although as I'm looking outside my window and it's gray and snowing, I wish I was actually there in Westwood instead of cold Chicago. So with that, I'll start. Today I'm going to talk about a little bit about the work that Shantanu hinted at with our app called BiAffect, but I'm going to focus mostly on some really recent work that we've been doing, looking at network alterations in patients with internalizing psychopathologies, that is patients with depression, depressive disorders, anxiety disorders, etc., and also to talk a little bit about implications for treatment based on these network alterations. So to start off is, here are my disclosures. The work I'm going to talk about was mostly funded by NIMH. So whenever I do a talk on brain networks or connectomics, I always like to start with a picture of this gentleman. If you've seen me talk before, you've probably seen this picture before. This gentleman is Sir John Batty Tuke. He was the president of the Royal College of Physicians in Edinburgh, and he was a psychiatrist in charge of one of the largest asylums in England. And he did a lot of really interesting clinical work, administrative work, and research in the area of psychiatry around the turn of the last century. And his work was summarized in this incredible compendium of lectures that he gave called The Insanity of Overexertion in the Brain. And this is a book that's actually freely available on Google if you want to look it up. But there's some really amazing insights that he came up with, before we had the technology that we have now to really understand the brain. And among those insights is a phrase that stood out to me, where he said that as yet the chemistry and mechanics of psychical processes are matters of surmise and theory. All we can say is that mental action is a function of connections. We have a right to infer that when the continuity of these connections is destroyed, interrupted, or structurally impaired, modification of function must ensue. So he had an understanding way back when of the importance of these brain network connections and how disruptions or alterations in these networks might give rise to some of the clinical phenomena that he was seeing and treating at the time. So this was back in 1894. 150 years later, NIH came to the same conclusion, saying that connectivity in the context of brain architecture has long figured in understanding, diagnosing, and treating certain neurological disorders. Increasingly disrupted or aberrant connectivity is being implicated or suspected in the etiology of disorders not previously considered from this perspective. And here you might think that they're referring to psychiatric disorders, but as I just told you, it actually has been previously considered from this perspective. Now we have better technology to make this consideration. And so, and that's what we do with the work that we've been doing over the last several years in connectomics, where the basic idea is to abstract the brain into a network or a graph, and then apply graph theory functions to characterize the brain as a network, and then characterize network alterations in the context of neuropsychiatric disorders. And some of the most common graph theory metrics that have been reported in the literature, or the simplest ones, are metrics like path length or network efficiency. And path length just describes the number of edges between any two given nodes in a network. So here between nodes I and J, the path length would be 3. The inverse of that is network efficiency. So longer path lengths means lower network efficiency. Another key metric that you'll see in the literature is something called nodal centrality, which describes the hub-like characteristics of a node in a network. And so if you look at the air transportation network, where I live in Chicago, Chicago O'Hare is a hub in the air transportation network, and where I would have been flying out of if I was going to be headed to LA to give this talk instead of doing it from home. So these two metrics are ones that are commonly described in the literature when people try to characterize brain network alterations in the context of various diseases. And in the context of mood and anxiety disorders, our lab and others have found alterations in these metrics. So things like reductions in global network efficiency associated with anxiety. Also, the reductions in network efficiency have been demonstrated in depression, and interestingly correlates with clinical symptom severity and cognitive measures like processing speed, such that lower network efficiencies are associated with slower psychomotor processing speeds. And then we and others have shown altered centrality or hub-like characteristics in brain regions involved in the default mode network, as well as limbic regions. And so a couple of years ago, I had the opportunity to work on an RDoC study, or a research domain criteria study, that was headed up by Dr. Luan Fan, who's now chair of psychiatry at Ohio State University. And what Luan was interested in was using the RDoC approach, which is a transdiagnostic approach, to understand treatment selection and the effects of treatment for internalizing psychopathologies like anxiety and depressive disorders. Given that there's a lot of heterogeneity within these disorders, and that the treatments that we use apply both to anxiety and depression, he thought that this was a good clinical target to understand some of these RDoC concepts, which try to get away from strict DSM-based categorizations of psychiatric diagnoses, and tries to look at what might be common across or transdiagnostically. And so this was a good sample to look at. And here is my grad student, Paul Thomas, who's an MSTP student getting his PhD in bioengineering. And he's been the one looking at the data, and it's his work that I'll be talking about the most this afternoon. So in this sample of patients, we had 69 patients compared to 24 controls. Here you see in this grid the distribution of diagnoses. So the majority of them tended to be more on the anxiety disorder spectrum versus mood disorders. And all of these participants were randomized to getting either therapy via cognitive behavioral therapy or medication treatment with an SSRI. I happen to serve as the study psychiatrist on the study providing SSRI treatment. In exchange for that, I was able to ask Luan to add various sequences to the imaging protocol and scales. And among those sequences that he added that he didn't originally have as part of the protocol was diffusion tensor imaging, which allowed us to look at structural brain network characteristics. And that's the first thing that Paul looked at in this sample, was to look at the structural brain network as defined by DTI to look at network-based alterations. And we decided to focus on the Uncinate subnetwork. These are brain regions in the medial prefrontal cortex, as well as limbic regions, which are linked by this association fiber, this white matter fiber, shown here in brown, and in yellow and magenta, we have the hippocampus here as a reference. And we wanted to look at the graph theory metrics for these nodes in the Uncinate network to see, A, are there differences between patients and controls? Are there network characteristics that are predictive of treatment response? And are there network characteristics that are predictive of treatment response in a treatment modality specific way that is specific to either getting therapy or getting medication? And one of the reasons why we focused on the Uncinate fasciculus was that there have been a lot of studies over the years showing abnormalities in this white matter tract in association with depression. So for example, evetylene has shown increases in white matter hyperintensities in this area. The white matter integrity of the Uncinate fasciculus has been associated with depression severity, as well as functional connectivity in multimodal imaging studies. And then people have even looked at other quantitative measures like fiber numbers showing that that's associated with depression severity. And our group and others have shown that, and there was a meta-analysis by Wen et al. That showed that a lot of studies have shown lower white matter integrity in depression in this, in the Uncinate fasciculus. So we thought this would be a good target to start with. And so the first thing that Paul found was that in general, looking at patients versus controls, there was a tendency for lower local efficiency, as well as clustering coefficient for the prefrontal nodes, and higher nodal strength and centrality for the limbic nodes in patients compared to controls. But one of the most robust findings we found was for the left medial orbital frontal cortex, where patients had lower local efficiency at baseline that also predicted their response to CBT treatment. So the lower their local efficiency at baseline, the more likely they were to respond to treatment, as well as the local efficiency increased with CBT treatment selectively, and it correlated with symptom improvement, shown here, where the change in local efficiency in this particular node was associated with change in symptoms as assessed by the Depression Anxiety Stress Scale, or DASS scale, which was our transdiagnostic scale that we used in this population. And what's nice about that finding is that it lined up with some previous work published by Helen Mayberg's group in the PREDICT trial, where they were also interested in neuroimaging predictors of treatment response to CBT and medication. And what they found was that functional connectivity between the subgenual cingulate cortex and the same left medial orbital frontal area distinguish who was going to respond to CBT treatment versus who was not going to respond to CBT treatment. So there was a nice alignment with this previous finding. All right. So that was a more hypothesis-driven approach where we focused on a specific subnetwork guided by what we knew in the literature. We also wanted to take a more unbiased, data-driven approach. And we also wanted to combine it with methods for discovering subnetworks of interest that might distinguish our groups of interest or might be associated with a variable of interest. And to do this, we looked at some clues from network theory that describe how does information spread across a network, right? So in this example, how do COVID-19 infections spread in a population? Or how does QAnon conspiracy theory spread on social media? It turns out that these diffusion dynamics, that is, or network diffusion dynamics, can be governed or understood with certain types of equations. And what we can use is something called the graph Laplacian, which is a special form of a network graph that is the difference between the degree matrix, and the degree just describes how many connections are linked to any given node. So here in this row 1 for node 1, the degree is 2 because it's connected to 5 and 2. And here in node 2 is connected to 3 nodes, 1, 5, and 3. And then you subtract the adjacency matrix, which describes the edges between each of these nodes, from the degree matrix, and you get this special matrix called the Laplacian matrix. And what's special about this matrix is that it actually encodes the diffusion dynamics along this graph. How does information flow, or how does heat flow in this graph? And that's described by this equation where the heat at a given node at a given time can be described by the eigendecomposition of the Laplacian matrix. That is the eigenvectors and the eigenvalues from this matrix. And so we can use this to characterize diffusion dynamics across the network. Another way to formulate it is to use a normalized version of that matrix to create something called the heat kernel, where every element of this heat kernel, which is a matrix that size node by node, each element encodes the amount of heat transferred between any two given nodes at a given point in time. And this is just shown graphically here. So if we start with heat at this node that's colored magenta, you can see that it will transfer to its neighboring nodes first before it extends to this other cluster on the right. And then at some point at time equal to infinity, you reach equilibrium where heat or information is distributed equally across all nodes. We can use eigenvectors of that Laplacian matrix to figure out what the eigenmodes are of the graph, and these eigenmodes characterize the diffusion dynamics across different time scales. So here, when we figure out what the eigenmode is, you can see that nodes that have values close together are the ones where heat or information will diffuse more readily across. If the values are very far apart, that means diffusion will take longer to reach those nodes. So basically, these eigenmodes encode the diffusion dynamics. And here's another example showing different eigenmodes. So this is the first eigenmode that was from the previous slide. And the first eigenmode, or the principal eigenmode, describes the diffusion dynamics over sort of the longest period of time. So as an example, if you were to start with heat at this node here, colored magenta, it would take these 4 time steps before you reach equilibrium. In the second eigenmode, which sort of splits the graph into core and periphery nodes, if you started heat at one of these core nodes, you actually reach equilibrium a little bit sooner. So these different eigenmodes capture diffusion dynamics across different time scales. And then we can actually sort of configure how these nodes relate to each other based on an eigenmode embedding. And that's just shown here where we're graphing it according to 2 eigenmodes. But we can do this for n number of eigenmodes for each node in the network. And again, the eigenmode encodes the ease of information transfer or heat transfer between those nodes. When you apply it to the brain, you see some interesting patterns. So in this principal eigenmode, you can see that there's a hemispheric split. So meaning that nodes that are similarly colored in red here have more readily information diffusion between them versus nodes that are differently colored here in blue. If you look at the second eigenmode, you see more of a medial lateral split. And in this third one, you see more of a dorsal ventral split. And these eigenmodes have been used to understand different neuropathological states. In one of the first papers to use this method in neuroimaging by Raj et al. That was published in Neuron, they found that the second eigenmode of network diffusion actually recapitulated atrophy patterns seen in patients with Alzheimer's disease, sort of reflecting the axiom from the Seeley paper that areas that are wired together, fire together, and expire together. We wanted to use this same method in our RDoC patients. And so what Paul did was he wanted to redefine how we looked at the structural connectome according to the eigenmode embedding using the heat kernel. But as I mentioned before, the heat kernel describes the diffusion dynamics at a given point in time. And he was faced with the challenge of, okay, what point of time do I sort of take my snapshot to look at the diffusion dynamics? So to do this, he was inspired by a paper by Abdul Noor et al. That was published in Neuroimage a couple years ago, where what they noticed was that if you actually look at the eigenmode embedding from the structural connectome or the heat kernel from the structural connectome, you can actually map it to the resting state functional connectome. You can find the parameters or the time point at which the heat kernel dynamics best reflect the pattern that we see in the resting state functional connectome. And so that's what Paul did to find the ideal time to sort of capture the heat kernel. And he did that for each individual subject in the study. And then this is summarized here, where basically we use the heat kernel from the DTI data. We find the time point by matching the best structure to function mapping. This creates eigenmodes for each subject. And then between each node, we can calculate a distance based on these eigenmodes. And that distance encodes the ease of information transfer or heat transfer between those nodes. And it's this distance matrix that we did our analysis on. And the analysis that he did was to use the network-based statistic to extract a subnetwork of interest. This is something that Zaleski published several years ago that's been widely used to identify subnetworks that distinguish groups of interest or to be correlated with a variable of interest. And the method is basically summarized here. We use the structure to function mapping to find our eigenmodes. We look at the distance between nodes in this eigenmode embedding to create the diffusion-based network embedding. We apply the network-based statistic to extract the subnetwork of interest. All right, so what did he find? So when he compared patients and controls, he found this fairly large subnetwork that connected prefrontal, insular, temporal areas where the distances were greater in the embedding, which meant that the diffusion was slower. When we did a deeper dive into this subnetwork to look at what might be some key nodes, there was one region in particular that had a high degree within the subnetwork, and that area, according to the Glasser parcellation from the Human Connectome Project, is area 8BM. And what's interesting about this area is that in a paper published last year, they found that this area is a core region in what they call the multiple demand system. And the multiple demand system is a network of frontal parietal regions that are activated in a task agnostic way. And they tend to be activated with tasks that require a huge cognitive demand. So it's a very important region in coordinating a lot of different cognitive activities. So it kind of makes sense that it popped up as a hub in this subnetwork that distinguished patients and controls. He also used a more coarser parcellation and found that in general, nodes that were in the anterior cingulate as well as the insula tended to be hubs in this subnetwork. So there was some overlap with the singular opercular network or the salience network. with this subnetwork. And then he also used multi-dimensional scaling to cluster the nodes in the subnetwork. And you can see at the top here in healthy controls in red that these nodes sort of represent this singular opercular or salience network. And that pattern tends to be disrupted in the patients where the cingulate nodes are now colored blue versus the more insular nodes. And the edges that connect these nodes are the edges where we see the slowest pattern of diffusion. He also looked at correlations with clinical symptoms and found that the heat transfer or the information transfer between the left anterior cingulate and the left posterior cingulate was actually positively correlated with depression symptoms, whereas other edges were negatively correlated, suggesting that the more depressed the participant was, the slower the information transfer was between these nodes. So this hub region and the left inferior frontal cortex and the left ACC and the left insula. He also looked at how it related to treatment response and found that the same connections that were associated with symptom severity were also associated with symptom response, treatment response, such that the worse off you were at baseline, the more likely you were to see a large decrease in your symptoms. And then looking at a treatment modality specific way, we actually ended up with different connections that were associated with CBT treatment response in terms of improvement in depressive symptoms. So information transfer between the right anterior cingulate and the right inferior parietal was negatively correlated with treatment response to CBT. And also the same hub region connected to the right superior parietal cortex was also negatively correlated with depressive symptom response to CBT. We also saw some correlations with change in panic symptoms with CBT treatment. The left ACC connected to the right lateral temporal region, and then the right ACC to the right superior parietal region were also significantly correlated with reductions in panic symptoms for those that got CBT treatment. We saw a different pattern with SSRI treatment that actually reflected sort of the overall treatment effects. Again, the left ACC to the left inferior frontal cortex was negatively correlated with depressive symptom improvements to SSRI treatment, and then left ACC to left insula information transfer was negatively correlated with reductions in depressive symptoms. So that's looking at how these network diffusion alterations respond to our traditional treatments. I sort of challenged Paul to come up with a way to see if we can find out where in the subnetwork is the optimal place to add heat to correct the diffusion deficits that we saw in the patient. So the idea is that we can possibly use this as a model for targeting a neuromodulatory treatment like rTMS, if we can find the optimal place in that subnetwork to stimulate to correct the network diffusion deficit. So what he did for each subject was he looked at the heat kernel for that subject and looked at the difference between the heat kernel for that subject and the average heat kernel for healthy controls as the standard. And that's shown here as XI, right? So XI is the difference between the heat kernel for a given patient and the average healthy control. And then what he wanted to figure out was how to minimize this difference if you added heat with this constant value of C to this matrix Y, which represents the node that you're adding heat to. And then you can iterate over all the nodes in the network and find out, okay, which node can I add the least amount of heat to minimize this difference? In other words, to correct the network diffusion deficits that we saw in the patient. And so when he did this over all the different subjects and all the different nodes in the subnetwork, he found that these these nodes were the ones that came up as solutions, with the anterior cingulate medial prefrontal node as the most common solution. And so if you were to select this node as the one to add heat to, which also, if you remember, is the sort of key hub node in this subnetwork, he was able then to compare the effect of adding heat to that node on the diffusion patterns for the edges within the subnetwork. So this is the subnetwork, and in blue, you're looking at edges where by t-test, we see a significant difference between patients and controls. So these are the deficits that we want to see if we can correct by adding heat to the node in the right anterior cingulate. And when he did that, he was able to correct most of the deficits shown here in light blue. There were a couple of edges where there was overcorrection, so increased heat compared to baseline. And that makes sense because these are the edges connected by the node where we added heat to. And then there were a couple edges that were unchanged. But for the most part, most of the edges were corrected just by adding the minimal amount of heat to this one key node. So to summarize, in internalizing psychopathologies, we see that network diffusion is selectively impaired in this prefrontal subnetwork with a hub in the anterior cingulate. That also happens to be this core component of the multiple demand system. And that these network diffusion deficits at baseline are correlated with symptom severity and maybe also predict treatment response to CBT and SSRI treatment. And by finding out where to add heat into the network to correct these diffusion deficits, this type of method may inform patient-specific targets for intervention. But there are a lot of, this is sort of brand new analyses, and there's a lot of sort of unanswered questions. And the first one that comes to mind is whether, is stimulation of a node with something like RTMS the same thing as adding heat in this model? And it might be the case. There are a couple of examples from Danny Bassett's lab where they use network control theory to suggest that there are aspects of the structural network topology that can inform stimulation targets in a predictable way to affect things like cognitive performance. There was also a paper published just this year where they showed that structural connectivity patterns actually predict TMS propagation as read out by EEG. And so perhaps this could be used to determine individual targets for neuromodulatory correction of these network-based diffusion deficits. And so that's something that we definitely want to pursue. All right, so I'm going to switch gears here. And I talked about network dynamics, but using structural brain networks. But of course, there's a lot of work being done looking at dynamics with functional brain networks derived from resting state fMRI, EEG, et cetera. This is something that Vince Calhoun has called the Connectome. And a number of studies have shown abnormalities in these functional dynamics in mood disorders. So, for example, Rosalind Kaiser has shown that there's altered dynamic functional connectivity between the medial prefrontal cortex and the default mode network. Others, including our lab, have shown that there are alterations in network dynamics associated specifically with rumination in the context of depression. And it's also been shown in bipolar disorder that these dynamic functional connectivity abnormalities are associated with cognitive impairments in the context of mood disorders. So we were interested in looking at this in EEG data. And I just wanted to throw up this picture just to show that the idea of looking at the dynamics is also not a new concept. So this is from William James, where he said thought is in constant change, and that what the change that he has particularly in view is that which takes place in sensible intervals of time, right? So paying attention to how thought changes over time. And that's what we wanted to do in this next study that I'm going to talk about, where we derived our connectomes from EEG data, and the edge weights are defined by a measure called the weighted phase lag index, which is a measure of coherence between any two given EEG channels. And we took a sliding window approach to look at how the EEG connectivity changes over time. We were inspired by work published by Maggie Xing, one of our former graduate students, where she was looking at these EEG connectomes in patients with social anxiety disorder. And she looked in different frequency bands and found a selective abnormality in the theta band such that patients with social anxiety had significantly higher theta network connectivity compared to controls. It also correlated with anxiety symptoms such that the greater the theta connectivity, the more anxious the patient was. And then there were also selective theta-based abnormalities in the graph theory properties of that network, exemplified by lower path lengths in the theta band for socially anxious patients. So we wanted to look at the dynamics of the connectome in the theta band in these patients while they were doing a particular task of relevance. In this case, the task was the emotion regulation task, where participants are asked to look at a neutral image like this teacup, or they're asked to look at a image that's designed to generate negative affect. And they're either asked to maintain that negative affect or try to use some kind of reappraisal technique to reduce the negative affect that they feel as a result of viewing the picture. And so my colleague Alex Liao came up with a really cool sort of data-driven approach to look at this dynamic data that we call the thought chart, where we basically take this sliding window approach as possible states, brain states, that the subject is experiencing while doing the task. And then we try to embed all of these states in this high dimensional space. And I'll summarize it here. So we have, from each subject, we have a snapshot of the connectome at different time points. Each of these connectomes form points in a S by T dimensional space, where S is the number of subjects, T is the number of time points. And then we look at the distances between these points in the high dimensional space, but we have to learn what the manifold is that the points lie on. So we use manifold learning methods and dimension reduction techniques to learn the manifold so that we can reconstitute the real distance between these connectomes shown by the curved line rather than the Euclidean distance, which is not correct in this high dimensional space shown by the straight line. Once we've learned the manifold, then we can represent the data in one of two ways. One, we can visualize it as a minimum spanning tree, which graphs basically a network of networks. So you can think of it as a meta network visualization tool. Or we could use nonlinear dimensionality reduction techniques to take it from this high dimensional S by T space into 3D space that we can actually visualize. So when we did this with our subjects doing the emotion regulation task, you can see here the minimum spanning tree shown here. And here you see the percentage of connectomes that belong to a particular part of the task, whether they're looking at a neutral stimulus, asked to maintain negative affect looking at a negative stimulus, or reappraise the negative affect looking at a negative stimulus. And you can see along the tree, there's a little bit of some distinction of what part of the task they're doing, but we see actually a better separation by subject, so that patients actually occupy mostly the left part of the tree and healthy controls the right part of the tree. As I mentioned, another way to visualize it is to use dimensionality reduction techniques to visualize it in 3D. And what we can do is we can actually reconstitute the trajectories of these time points in this three-dimensional space. And what you're seeing here are streamlines that represent the positions of these connectomes in 3D space. And when you take a top-down look at these trajectories, you can kind of see how they occupy different parts of this space. So healthy controls connectomes when they're looking at neutral stimuli is here. When they're doing the maintain part of the task, it's located here. When they're doing reappraisal, it's here. Social anxiety patients occupy mostly the right side of this space. And you can also see that the trajectories occupy a larger sort of volume in this space, and we can quantify that as well by looking at the lengths of these trajectories in healthy controls versus social anxiety patients. And when we do that, we can see that when we look at the neutral part of the task, social anxiety subjects have really high trajectory lengths. It's high during maintain, and it remains high during reappraisal. And so we think that this increase in trajectory length during the task represents the cognitive demand of the task. If you look at healthy controls, it sort of increases as you go from neutral to maintain to reappraise. So this is increasingly cognitively demanding as you go from these three steps, whereas the social anxiety patients already start high. And sort of as further evidence of that, we could look at the tendency to use some of these emotion regulation techniques in real life by looking at their ratings on the emotion regulation questionnaire. And what we found was that patients that or participants that tended to use reappraisal as a day-to-day emotion regulation strategy tended to have lower reappraisal trajectory lengths. So we think that it might have to do with the cognitive effort involved in this emotion regulation task. Another example is from a study where we applied the same thought chart approach to participants who did different types of meditation practices. And we can see that the minimum spanning tree actually segregates connectomes that are recorded during different types of meditation practice. And we saw some significant correlations with changes in trajectory length and mindfulness, such that the more mindful a person was, the more they were able to sort of reduce their trajectory lengths or slow down their mind. Because the other way to think about the trajectory length is that since it's a length over a particular period of time, we could also think about it as sort of speed of thought, where shorter trajectory lengths might represent slower thought processes. And that also, the idea of thought processes and speed was theoretically described in a paper by Pronin and Jacobs, where they tried to characterize different aspects of psychopathology along axes of speed of thought and variability of thought. So if you have low variability but high speed, that's more like anxiety. Low speed of thought and low variability, depression. High speed of thought, high variability, mania. All right, so from our dynamic EEG network analysis, we see that there are altered theta band network properties associated with anxiety, and that we think that the dynamics in the theta band might represent cognitive effort during emotion regulation. And so one question I had was whether we can, similar to the story with our network diffusion alterations, can these altered network properties serve as a target for intervention? And we've done a pilot study using transcranial alternating current stimulation, where we get a resting state EEG. We find out where do we see the highest theta connectivity, and use those channels as a target for transcranial alternating current stimulation, where we're stimulating out of phase to reduce what we consider abnormally high theta connectivity in the context of anxiety. And this is the study design. We had people randomized to getting TACS, which is our sort of active condition, and TDCS, which is our active control. And even within both stimulation paradigms, subjects were randomized to getting sham or active stimulation during the emotion regulation task or during a resting state EEG scan. And we have a number of outcome measures that we looked at. One was one is the actual negative affect that the participant is experiencing during the task, as well as measures of depression and anxiety. And then we have EEG related outcome measures looking at the effects of stimulation on our EEG readouts, whether it's a late positive potential or whether it's EEG based Connectome measures. The Connectome measures and EEG measures are still a work in progress, but we have some data showing that TACS selectively reduced negative affect particularly during the reappraisal part of the task. We did see that same effect with TDCS. And then we also saw a reduction post-stimulation in depressive symptoms in the TCS arm, but not in the TDCS arm. Now, whether these reductions in negative affect and depression ratings are related to actually changing theta connectivity is still an open question. And this is work done by my other graduate student, Jessica McAleer. All right, so when looking at connectomes and emotion regulation, we see that dynamic EEG connectome properties characterize emotion processing and emotion regulation, and it may serve as a target for network-based intervention with a modality like transcranial current stimulation. So in addition to the network of brains that I've been talking about, we also consider the network of connected devices that we are all linked to, like our smartphones and our smartwatches. And so I'm just going to end in the next couple minutes talking about a couple of ongoing studies that we have now where we're trying to incorporate some of our neuroimaging methods into our understanding of digital mental health, whether digital mental health interventions or digital mental health technologies for tracking symptoms. The first study that we have ongoing is called UnMASCK, which stands for Unobtrusive Monitoring of Affective Symptoms and Cognition Using Keyboard Dynamics. This is related to what Shantanu was talking about in his introduction. My colleague Alex Liao developed an app called BiAffect, which replaces the default keyboard of your smartphone with a customized keyboard that allows us to track how you're typing. And we've shown that these typing dynamics are associated with clinical symptoms of depression and mania. We've also have some preliminary data suggesting that it's also associated with cognitive measures like processing speed and executive function. And so what we wanted to do in this study was link what we measure on the phone to what we can measure in the brain, and to see to what degree do our typing dynamics track with connectomic correlates of cognitive dysfunction in the context of mood disorders. So we're recruiting across the spectrum of mood disorders. So it includes bipolar patients, patients with unipolar depression, dysthymia, and we also have some controls as well. And we have them, after doing a baseline assessment, we have them using the app for a couple of weeks. They come in for a scan, more clinical and cognitive assessments, and then we repeat that for another 2 weeks. So we can look to see to what degree does what we measure with the phone link to what predict what we can see in the brain, and vice versa. And here's just an example of some of the hypothesized links between the keyboard metric shown here as the BiAffect metric. So we think that typing speed, we've shown that it's associated with psychomotor processing speed, and based on our work and others, that processing speed is linked to measures like brain network efficiency, as an example. The other study that we have going on is in late life depression called Rembrandt, where we're interested in trying to figure out what are the markers that we can use as a predictor of relapse for patients who have successfully been treated for their depression. Because first of all, in late life depression, it's very hard to treat. And even if you do get people well, it's hard to keep them well. There's a very high relapse rate on the order of 40% within 2 years after reaching remission. So the goal is to link functional network markers, things like network instability in cognitive control networks, to actual neuropsych assessments of cognitive function, and see how those relate to possibly in a predictive way to worsening clinical symptoms. And so we are using multimodal neuroimaging, as well as mobile cognitive assessment, ecological momentary assessment, to make these links. And here's the overall study design. So we're recruiting people who are currently in remission, or if they're actively depressed, we treat them to the point of remission, and that's when they enter the study. We follow them longitudinally for 2 years. with MRI scans every 8 months. And for some of the participants, we're actually going to be doing longitudinal sampling of ecological momentary assessment measures, as well as mobile cognitive assessment measures across the 2-year period. And here's just a sample of some of the mobile cognitive tests. So they're basically tests of working memory, attention, concentration, and processing speed. And here's the imaging protocol. We actually have added DTI to this, but it's a pretty straightforward imaging protocol. And if you want to know more about the theoretical basis for the study, we published a paper a couple years ago in the American Journal of Geriatric Psychiatry describing the model. And then the last study that we've just got started this year is a digital mental health intervention called Speak, which is a problem-solving therapy coach that works on the Alexa platform, designed for patients with mild to moderate depression and or anxiety. Basically, it resides on a smartphone or an iPad. We utilize Alexa's natural language understanding to deliver the therapy sessions, and we deliver 12 sessions over 8 weeks. And we're neuroimaging, oh, sorry, and I'm just going to play a quick sample of what it sounds like. Welcome to your first counseling session. If you could hear that. Please make sure
you the voice actually faded. Olu, can you please? And yeah.
Yeah, I'll skip it. But basically, it sounds like Alexa as a therapist. And so where neuroimaging comes into play is that we use the R61 mechanism, which means that we have to show target engagement for the intervention. And in this case, the target are actually neural circuits that have been previously been shown to be associated with treatment response to problem-solving therapy. So this involves amygdala, insula activity in response to a non-conscious conscious threat task, as well as dorsal ACC activity and connectivity in the context of a go, no go task. And so those are the targets for the treatment. And we'll be comparing those who get the app to a waitlist control on the intervention's effects on those targets. All right, so I will wrap up here to leave a couple minutes for questions. I want to acknowledge Alex, who's my co-pilot for the lab, and thank you for your attention.
It's an excellent talk, Olu. Very inspiring. So any questions from the audience? If you have any questions, please unmute yourself and ask them. But before we do that, as you said, Olu, I just want to re-acknowledge Sarah Chang, who actually suggested you as a speaker, and of course, Catherine, whom you interned. I didn't, not intern, you did research with. Yeah, I forgot to mention that at the beginning. So, hey, thanks for reminding me. So yeah, please go ahead and unmute yourself and ask any questions if you have them.
Hi, Olu. Thank you so much. That was a brilliant talk. So you're doing such innovative work and so sophisticated and advanced. It's really amazing. I have many, many questions. I don't even know where to start. You did such a great job of explaining the methods, but I didn't really understand the mapping of the heat connectome to the resting state data. So maybe you can just mention that again. I also wanted to know, sorry, let me just finish with my questions so that I can get them all out and you can address them in order.
Okay.
What do you make of the mostly rightward subnetwork that was extracted, and what does that mean for brain stimulation? So does that mean we should be stimulating like right-sided anterior cingulate? Gosh, what were the other questions? They're just transcranial alternating current and TDCS. So that's just at one time point. I didn't, or that is repeated treatment over time. I didn't get that.
So one time.
Anyway, one
yeah.
time.
So
Okay.
So I'll do the last one first, and that's the easiest one. So that was one time. And so one of the things that we're trying to figure out is that if this were to be translated into a treatment, how many times does somebody need to be stimulated for you to have sustained effects? It's still unclear for us. So that's one of the things we want to try to figure out. Then your second question was about the laterality of the subnetwork, and I'm not sure yet. What I think is more potentially powerful about this approach is the idea that you would be able to find a unique solution for a particular individual. So it may not always be on the right, it may be on the left, but the idea is that it would be patient specific. And then your first question was about the structure to function mapping. That's really taken from that Abdul Noor paper in Neuroimage, where basically you're solving for parameters where you get the best fit between the heat kernel and the resting state functional connectome. There are several parameters in the equation that represent time, the hypothetical time step.
Right, but why make that mapping? Because I mean, sure, that addresses one part of the connectome, but just the structural connectome that doesn't, the heat connectome that doesn't necessarily map is also surely relevant, right? I don't know. I need to think about it.
Yeah, I mean, because basically you can calculate it at different sort of hypothetical time steps. So the question is, what time step was the best? Right. And so the idea behind the structure to function mapping is to pick the sort of ideal time step for that particular subject's heat kernel.
Got it. Very, very interesting. I'll let others ask questions now, but thank you. That was just excellent.
Thanks.
Hey Olu, this is Sarah Chang. I was previously in Lee Williams' lab, if you remember me.
I remember, yes.
One of the things that I'm really interested in is motion regulation. So I was pulled in particular with your 3D streamlines and the trajectory length part. And I was wondering how you were interpreting the lack of difference between groups in trajectory length for reappraisal. I know you talked a little bit about the trend, but
right. So I think
could...
So I think the way I've interpreted it is that basically, if it represents some kind of cognitive effort, there's sort of like a ceiling effect, and that the socially anxious folks already start up revved up really high, and they can't really go much higher. Whereas healthy controls, as the sort of task demand increases, you see this stepwise increase in the trajectory lengths accordingly. So basically, the socially anxious people are start high, stay high. And so the differences that we see are in the neutral and maintain condition, not in the reappraisal condition.
Got it. I guess the way that I was interpreting it initially was that the processing between groups for reappraisal was about the same, which was a little bit surprising to me.
Yeah. I mean, I think if we look at, in this particular sample, if you look at the success of their reappraisal, that is, if you look at their negative affect ratings during reappraisal, there wasn't a difference between patients and controls.
Got it. That makes a lot of sense.
Yeah.
But yeah, great talk. It was super interesting. I wish we were able to host you and talk a bit more.
Yeah, well, I'm still open for a trip to LA, especially now.
we'll make that happen. But right now, nobody's in the lab, so you'd be there on your own.
Okay, yeah, it's kind of like our lab.
Yeah.
Any other questions?
Hi, Dr. Ajilore. Am I pronouncing your last name correctly?
Yes, you are. Thank you.
Awesome. I am an undergrad here in the neuroscience department, and I just had a question more generally about kind of where you see this treatment possibly going. Because when I'm like reading about or listening to you talk, which is awesome, even though I didn't understand some things for sure, thinking about implications of treatment, like having this accessible to so many different people and being able to individualize treatment for mental health and just like that sounds pretty awesome. But did you have any thoughts about that and like future implications?
Yeah, I mean, I think that's the way things are going, right? Is there's a lot of heterogeneity in how we diagnose folks, and a lot of trial and error in how we treat them. And so, I think the great potential of this kind of work and technology is to be able to have more precision, in how we diagnose, precision in how we treat. So I think that's where things are going. I mean, there's some really cool work out of Stanford where they use, functional neuroimaging to guide intermittent theta burst stimulation treatments, and shown that people who have failed treatments like ECT or other RTMS treatments actually respond if you use a more specific targeting mechanism for that patient.
Wow, thank you so much. Yeah, this is super, super awesome. So I'm hoping to hopefully see it used more often in the future.
I think we're getting there.
Is there time for another question?
Yeah, absolutely.
Hi, Namthia Sivana. Nice to meet you over Zoom. I had a question that maybe I missed about the theta findings. We have a trial targeting amygdala theta in PTSD patients. So, of course, I'm very intrigued by this finding. And I was wondering if you could comment on the sort of regional localization across participants and any differences across different types of negative valence,
Yeah, so that's a good question. Unfortunately, we didn't have a lot of channels. So I'm not confident how well we could do any kind of source localization with the EEG data that we have in the TCS pilot study. But I think it aligns with previous work that showed basically sort of increased theta. So there's animal work that shows increased theta coherence between the medial prefrontal cortex and the ventral hippocampus. That's been also seen to some degree in human studies, like with MEG. And so I think that I don't know if we're targeting that specifically, but I think that is the target that we do want to hit. I don't know if that answers your question.
Yeah, no, that's actually what I was thinking you'd say. So it's great to hear that.
Yeah.
Thanks for that. And in terms of patient types, are these like generalized anxiety? Or have you looked at
yeah. So for the TCS, we actually took a transdiagnostic approach as well. So so there's anxiety disorders, mood disorders. I think the only thing we excluded was bipolar disorder, but you could have MDD or generalized anxiety.
Great, thank you. Great work. Thank you.
Thanks.
We have time for one more question, since this is on Zoom and people are not traveling. And I have one while others are thinking. So the heat kernel, it's very interesting. Sorry, is someone speaking? Okay, so it's nice to see the heat kernel and the Laplacian matrix being applied to segmentation and computer vision problems now being applied here. I'm just wondering, when you talk about the transfer of heat from one node to the other and this kind of a dynamic connectome, have you just thought about kind of a control steering approach to actually steer the nodes over time to maybe improve therapy or something.
Wait, so I missed part of what you said.
So can you use these nodes and the heat, for instance, in a control theoretic sense to actually steer?
Yeah, so that's similar to that's similar to the work from Danny Bassett's lab. It sounds like where basically you pick nodes that have high modal controllability, right, that allow you to sort of change states.
Yes.
Right. Yeah. So that's very much aligned with what she's demonstrated with her work. Yeah.
And you actually have data for that because you are showing these trajectories of different states.
No, we haven't. So we haven't linked those two. Yeah.
Okay.
We haven't linked those two.
Okay.
But now you've given me an idea, though.
Okay. So great. Right. And also then really quick, when you change the heat at different nodes, did you actually even try to do a simulation where you observe the correlations with the mood? If they go up and down.
Oh, that's interesting. Yeah, no, we haven't done that yet, right? To see if it actually changes the relationship with the clinical symptoms.
Yeah.
And yeah, that's actually a good question. No, we haven't looked at that yet.
Okay. So time for one more question, and then we'll let Olu go. Okay, so Olu, it's really great talk, and as Catherine said, we should probably have you here in person in the future.
Great.
And nice to hear all these nice different approaches, traditional mathematics, and actually being really nicely used. And I've seen methods targeting these specific things like heat kernels and Laplacian matrices, eigenvalues, but never so comprehensively explained in one experimental study design and applications to neuroimaging. So it's very nice. Thanks.
Yeah, thanks. It's sort of the magic. Alex has the strong math background, and I think about the application, so it works well.
Great. so let's thank Olu again, and be safe.
Thank you.
Seminar. Bye.
Take care.